Welcome to the deep dive. Today we are plunging into, well, the absolute bedrock of biology, the
A
cell.
fundamental unit of life,
Yeah.
capable of independent existence, doing all the essential things life needs.
Exactly. We've got a pile of sources here. Articles, notes, research bits covering pretty much everything from cell structure and function to how we even figured out what cells were.
And our mission today, basically, to sift through this, pull out the really key insights, and give you a solid handle on what makes a cell, well, tick.
Highlighting some fascinating details along the way, hopefully.
Oh, definitely. Think of it like a focus briefing on this tiny world that makes up, well, everything alive.
Okay, let's get into it. The first big idea these sources really drive home is that basic difference between living and non-living things.
And the heart of that difference, yeah, it's the cell. The presence of cells is what defines life.
Every living thing, from a single bacterium to us, made of cells.
A product of cells, right?
So the cell isn't just like part of life, it's the basic unit itself.
Precisely. Look at single-celled organisms.
Mm.
It's incredibly clear there that one cell does everything needed for life, feeding, growing, reproducing,
Responding to its environment.
all of it.
Hmm.
And the sources make a strong point. Anything less than a whole, complete cell just can't manage independent living. You need the full package.
It's kind of mind-blowing that something so small can be so self-sufficient. How did we even start seeing this world?
Well, it took curiosity and, crucially, the right tools. People like Anton von Leeuwenhoek.
With his early microscopes.
Exactly. He was the first to actually see and describe living cells, those little animalcules moving around.
Wow.
And then later, Robert Brown spotted that denser bit inside plant cells, the nucleus.
So technology was key. Bet our microscopes meant deeper understanding.
Absolutely. The evolution of microscopes, especially leading up to the electron microscope in the 1950s, just blew the doors open. Suddenly, we saw intricate details, not just blobs.
And all those observations eventually led to a huge idea, cell theory.
A real cornerstone of biology, yeah. And it built up over time. It really got going in the 1830s. You had Matthias Schleiden, a botanist.
Right, he said all plants are made of cells.
And almost at the same time, Theodor Schwann, looking at animals, reached a similar conclusion. Animal bodies are also made of cells.
He also spotted that outer layer on animal cells, the plasma membrane.
And notice plants have the cell wall outside that.
Right. So they put together the first version. Plants and animals are made of cells and their products.
But there was still a big question mark.
Mm-hmm.
Where do new cells actually come from?
Ah, that's where Rudolf Virchow comes in, 1855. Omnis cellula e cellula.
Which means...
All cells arise from pre-existing cells.
Exactly. That was the missing piece. Cells divide, they don't just pop into existence. That completed the modern cell theory.
So the modern theory basically has two main points.
One, all living organisms are made of cells and stuff cells produce.
Okay.
And two, all new cells come from cells that already exist. Simple, but incredibly powerful.
And understanding cells at this level lets us do things like what the sources call reductionist biology.
Yeah, this is fascinating. It's about applying physics and chemistry to biology. You take complex living things, tissues, organs, and break them down.
Analyze the chemicals, study the molecular processes.
Right. Sometimes even using bits of cells or systems without any cells at all.
So it's like understanding a machine by looking at its individual parts and how they work according to physical laws.
Yes, that's a good way to put it.
Ah,
By understanding the physics and chemistry inside the cell, you can explain really complex stuff, muscle contraction, memory, even what goes wrong in diseases at a molecular level.
it connects the small scale to the big picture.
Exactly.
All right, so with that theory established, let's look at the general layout of a cell, like what you'd see looking at an onion cell or a cheek cell.
Okay. You see that outer boundary, the rigid cell wall in plants,
Mhm.
or the more flexible cell membrane in animal cells like our cheek cells?
And inside that,
You've got the cytoplasm. It's this sort of semi-fluid, jelly-like matrix filling the cell.
kind of the cell's main arena where lots of chemical reactions happen.
That's right. And then in many cells, you see a distinct, often central structure, the nucleus. It's got its own membrane around it.
And inside the nucleus is the important stuff.
The genetic material, the DNA, organized into chromosomes.
Mm-hmm.
And whether or not a cell has this membrane-bound nucleus is the big divider.
Ah, leading to the two main types,
Yeah.
eukaryotic and prokaryotic.
Exactly. Eukaryotic cells do have that membrane-bound nucleus. That includes protists, plants, animals, fungi, basically most life forms we see easily.
And prokaryotic.
They lack a membrane-bound nucleus. Their DNA is just sort of floating in the cytoplasm. These are things like bacteria, blue-green algae, mycoplasma, PPLO, simpler structures generally.
Eukaryotes also seem to have a lot more internal compartments, right? Organelles.
They do. That's a key feature. Lots of membrane-bound organelles like the ER, Golgi, lysosomes, mitochondria, vacuoles. Prokaryotes mostly lack these complex internal membranes.
Is there anything both types share structure-wise?
Yes, absolutely. Ribosomes. Crucial little things. The protein factories.
Precisely. And they are not enclosed by a membrane. You find them in all cells, prokaryotic and eukaryotic. In eukaryotes, they're in the cytoplasm, on the ER, even inside mitochondria and chloroplasts.
And animal cells also have centrosomes for cell division?
Correct, another non-membrane-bound structure. It really shows, as the sources emphasize, the incredible diversity of cells. Size, shape, what they do.
It's wild. You've got mycoplasmas, tiny, like 0.3 micrometers, then bacteria, a bit bigger.
Then jump to an ostrich egg, technically one huge cell. Or human red blood cells about 7 micrometers across. Nerve cells can be super long.
Shapes too, discs, columns, threads, totally irregular.
And the shape nearly always relates to the function, you know? A long nerve cell makes sense for sending signals.
Okay, let's focus on those prokaryotes first. Bacteria, cyanobacteria. They seem simple, but they're everywhere.
They are incredibly successful. Usually smaller, they multiply much faster than eukaryotes. Even though bacteria come in different shapes, rods, spheres, spirals, their basic internal plan is quite similar.
What's that fundamental setup? Most have a cell envelope, usually cell wall outside the plasma membrane, that gives shape and protection. Mycoplasma are the exception. No cell wall.
And inside.
You have the cytoplasm. And the genetic material, typically one circular chromosome of DNA, is just there in the cytoplasm, not wrapped in a nucleus. We call it naked DNA.
And lots of bacteria have those extra little DNA circles, plasmids.
Yes, plasmids. Small, circular DNA, separate from the main chromosome. They often carry useful genes, like for antibiotic resistance. Huge clinical importance. And they're key tools for genetic engineering.
Any unique prokaryotic structures inside?
A really interesting one is the mesosome. It's an infolding of the plasma membrane.
What does it do?
The sources list several functions. Helps make the cell wall, helps copy and divide the DNA involved in respiration, secretion, plus it increases the membrane surface area.
And some can photosynthesize.
Right, like cyanobacteria. They have internal membrane extensions called chromatophores, which hold the pigments.
How do they move around, if they do?
Motile bacteria often use flagella. These are thin filaments sticking out. But a bacterial flagellum is structurally very different from a eukaryotic one. It rotates more like a propeller.
Simpler mechanism?
Simpler, but effective. Built from a filament, a hook, and a basal body anchoring it.
What about other things on the surface? Chelae and fimbrio.
Ah, yes. They are not for movement. Chelae are usually longer, tubular, sometimes involved in transferring DNA between bacteria. Fimbria are shorter, like bristles.
What are they for then?
Attachment, sticking to surfaces, rocks, host tissues, you name it. Really important for bacteria that cause infections, for example.
And the ribosomes, you said they were different.
Smaller than eukaryotic ones. We call them 70S ribosomes, made of 50S and 30S subunits. Remember, S is Svedberg unit, about sedimentation, not simple addition.
Still making proteins, though.
Still the protein factories, yes. Often you see several ribosomes attached to one messenger RNA molecule, churning out proteins. It's called a polysome.
Any storage bins inside prokaryotes?
Yes, inclusion bodies. They store reserve materials right there in the cytoplasm. But, and this is key, they are not membrane-bound, just granules lying free.
Like what?
Phosphate granules, glycogen, even gas vacuole in some photosynthetic types to help them float.
Okay, fascinating stuff. Let's switch over to the eukaryotes then. Protists, plants, animals, fungi, more complex.
Much more complex, largely because of that internal compartmentalization, all those membrane-bound organelles creating specialized environments within the cell. Plus, the organized nucleus, complex cytoskeleton.
And you mentioned key differences between, say, plant and animal cells.
Absolutely. Plants have the cell wall, plastids like chloroplasts, and usually that big central vacuole. Animals generally don't.
But animal cells have centrioles.
Right. Usually involved in cell division.
Yeah.
And mostly absent in plant cells.
Let's walk through some of these eukaryotic organelles, starting with the boundary, the plasma membrane.
Okay. This isn't just a static bag. We really started understanding its details with the electron microscope in the 50s. Chemical studies, especially on red blood cells because they're easy to get, showed it's mostly lipids and proteins.
The main lipids being phospholipids.
Yes, arranged in that famous bilayer. The heads face the watery environments inside and outside the cell. They're hydrophilic. The tails are hydrophobic. fatty acid chains pointing inward, away from water. Cholesterol is also tucked in there, affecting how fluid it is,
And proteins are mixed in?
embedded in or associated with that bilayer. The ratio varies. Red blood cells are about 52% protein, 40% lipid. Some proteins just sit on the surface, peripheral. Others are partly or fully buried, integral. Some even cross the whole membrane.
Which brings us to that fluid mosaic model.
Yes, Singer and Nicholson, 1972. Still the best description. It sees the membrane as a fluid mosaic. The lipid bilayer is like a somewhat fluid sea, and the proteins are like icebergs floating or moving laterally within it.
And this fluidity isn't just incidental.
Oh no, it's crucial. Cell growth, forming junctions between cells, secretion, bringing things into the cell, endocytosis, cell division, all these depend on the membrane being dynamic and fluid.
And it controls what gets in and out, right? Transport.
It's selectively permeable. Small, uncharged things can sometimes diffuse right through. Water moves by osmosis. But charged ions or larger polar molecules like glucose need help.
How do they get across?
Sometimes through channel proteins or carrier proteins that facilitate their movement down the concentration gradient. That's facilitated transport, passive, no energy needed.
But what if the cell needs to move something against the gradient, like pumping waste out?
Then it needs energy, usually from ATP, that's active transport. Carrier proteins act like pumps, forcing molecules across even if they're moving from low to high concentration. The sodium-potassium pump is a classic example.
Okay. Now, outside the membrane in plants and fungi, there's the cell wall.
A non-living, fairly rigid layer. Gives shape, protects the cell from damage, prevents it from bursting if it takes in too much water, and is involved in cell-to-cell interactions.
What's it made of?
Varies a bit. Algae might use cellulose, galactans, mannans, even minerals like calcium carbonate. Other plants mostly use cellulose, hemicellulose, pectins, and some protein.
And it's not static, it forms in layers.
Right. Young, growing plant cells have a flexible primary wall. As the cell matures, a thicker, stronger secondary wall can be laid down inside the primary one. And neighboring plant cells are stuck together by the middle lamella, mostly calcium pectate, like a glue.
Are the cells totally sealed off them?
No, they have connections called plasmodesmata. These are channels through the walls and middle lamella linking the cytoplasm of adjacent cells. Allows for communication and transport between them.
Right. Now, inside, you mentioned that coordinated system, the endomembrane system?
Yes, a group of organelles working together. The endoplasmic reticulum, ER, the Golgi complex, lysosomes, and vacuoles. Their functions are interconnected, especially in producing, modifying, and transporting proteins and lipids.
But mitochondria and chloroplasts are separate.
Their functions aren't directly coordinated in that same production line way, so they're usually considered separate from the endomembrane system.
Got it. First stop, the ER.
Endoplasmic reticulum, a huge network of interconnected membranes, tubules, and flattened sacs, called cisternae, spreading through the cytoplasm. It creates an internal compartment, the lumen, separate from the main cytoplasm.
And there are two kinds, rough and smooth.
Exactly. Rough ER, our ER, looks rough because it's studded with ribosomes. It's heavily involved in making proteins that will be secreted or sent to other organelles and also in folding them correctly, often connected to the nuclear envelope.
And smooth ER.
No ribosome, so it looks smooth. It's big in lipid synthesis, including steroid hormones, also detoxifies drugs and poisons, and stores calcium ions.
Next up, the Golgi apparatus, named after Camillo Golgi.
Yep. Looks like a stack of flattened membrane sacs, the cisternae. It has a receiving side, the cis's face, usually near the ER, and a shipping side, the trans face.
What's its main job, like a processing center?
Sort of like the cell's post office or finishing factory. Vesicles carrying proteins and lipids arrive from the ER at the cis face. As they move through the Golgi stacks, the contents get modified, sorted, and packaged into new vesicles that butt off the trans face.
Modified how?
Sugars might be added to make glycoproteins or glycolipids, for example. It ensures things are correctly addressed and sent to their final destination.
Okay. And then lysosomes. They sound destructive.
They are the recycling centers or maybe the demolition crew.
Yeah.
Membrane-bound sacs formed by the Golgi, packed with hydrolytic enzymes that break down molecules using water.
Like what kinds of molecules?
Proteins, lipids, carbohydrates, nucleic acids. They work best at acidic pH, which is maintained inside the lysosome. They digest stuff brought into the cell or break down old, worn-out organelles. Very important for cellular housekeeping.
Last in this system, vacuoles.
Membrane-bound sacs, basically.
Yeah.
Membrane is called the tonoplast. It can hold water, sap, waste products, pigments. Depends on the cell type.
Plants have that huge central one, right?
Yes. Can take up 90% of the cell volume. crucial for maintaining turgor pressure, the water pressure that keeps plant cells firm. The tonoplast pumps ions into the vacuole, drawing water in.
Do other organisms use them?
Oh, yes. Amoeba has a contractile vacuole for pumping out excess water. Many cells form food vacuoles by engulfing food particles.
Okay, moving beyond the endomembrane system, the power generators.
Mitochondria.
The powerhouses indeed. Where aerobic respiration happens, generating most of the cell's ATP energy currency, their number varies a lot. High-energy cells like muscle have tons, others fewer.
And the distinctive double membrane structure.
Right. A smooth outer membrane and a highly folded inner membrane. Those folds are called cristae.
Why folded?
Increases the surface area dramatically. The inner membrane is where the electron transport chain and APP synthesis happen, so more surface area means more energy production capacity.
And the space inside the inner membrane.
That's the matrix. It contains mitochondrial DNA, a small circular chromosome, interestingly, plus 70S ribosomes, similar to prokaryotes, and enzymes for the Krebs cycle. So they can make some of their own proteins.
They can. And they divide by fission, somewhat independently, all hints about their ancient origins, likely as free-living bacteria.
Fascinating. Now, in plants and some protists like Euglena, we also find plastids.
Right, large organelles, often easily visible. Classified based on pigments. Most famous are chloroplasts.
Site of photosynthesis, the solar panels.
Exactly. Contain chlorophyll, green, and carotenoid, yellow-orange pigments to capture light energy. Also double membrane bound.
What's inside them?
An inner space called the stroma. Within the stroma are flattened sacs called thylakoids, often stacked into grana. The light reactions happen on the thylakoid membranes. The sugar making happens in the stroma.
And like mitochondria, they have their own DNA and ribosomes.
Yes, small circular DNA and 70s ribosomes, again pointing to an independent origin.
What are the other plastid types?
Chromoplasts contain carotenoids, giving flowers and fruits their yellow, orange, red colors. Leucoplasts are colorless, for storage. Anelloplasts store starch, like in potatoes. Eleoplasts store oils. Aleuroplasts store proteins.
Okay. And ribosomes, we keep mentioning them. Just to recap their basic structure in eukaryotes.
First seen as little dots by George Palade, made of ribosomal RNA and protein, no membrane. Eukaryotic ones are ADS type, larger than the prokaryotic 70S, made of 60S and 40S subunits.
Still the protein builders.
Universal protein synthesis machines.
Now, what holds the cell's shape and allows movement inside? The cytoskeleton.
Yes, an intricate network of protein filaments throughout the cytoplasm. Includes microtubules, microfilaments made of actin, and intermediate filaments.
What does it do? More than just scaffolding?
Oh yeah, provides mechanical support, maintains shape, allows cells to move, and also acts like internal railway tracks for motor proteins to move organelles and vesicles around the cell. Very dynamic.
And for external movement, some eukaryotes use cilia and flagella.
Hair-like extensions. Cilia are short, numerous, often beat in coordinated waves like oars. Flagella are longer, usually just one or a few, use a whip-like motion for propulsion.
But structurally different from the bacterial flagella.
Very different. Eukaryotic ones are covered by the plasma membrane. Inside is a core called the axonome, with that characteristic 9+2 arrangement of microtubules, 9 pairs around the outside, 2 single ones in the center. This structure allows for bending.
And they grow out from.
Basal bodies, which
Yeah.
are structurally pretty much identical to centrioles.
Which brings us to the centrosome and centrioles, found in animal cells mostly.
Yes. The centrosome is the main microtubule organizing center in animal cells. It usually contains 2 centrioles, arranged perpendicular to each other, embedded in amorphous material.
And the centrioles themselves have a specific structure.
A very distinct cartwheel structure,
Yeah.
made of 9 triplets of microtubules arranged in a cylinder. No central microtubules like in cilia flagella.
What are their jobs?
They form those basal bodies for cilia and flagella, and crucially during animal cell division, they organize the mitotic spindle, the fibers that pull the chromosomes apart.
Okay, almost there. Let's go back to the control center, the nucleus.
The big boss, first described by Robert Brown, contains the cell's main genetic library, enclosed by the nuclear envelope.
That double membrane.
Yes, 2 membranes with a space between. The outer membrane is continuous with the ER, and it's dotted with nuclear pores.
Which act like gateways.
Exactly. They regulate what gets in and out. RNA needs to get out. Proteins needed for DNA replication or transcription need to get in. Very selective.
Do all eukaryotic cells keep their nucleus?
Most do, but some, like mature red blood cells in mammals or sieve tube cells in plants, lose theirs. Which raises that interesting question you mentioned, are they fully living without that control center? They function, but can't maintain themselves long-term or reproduce.
A philosophical point almost. What's inside the nucleoplasm?
You've got the chromatin, that's the DNA complex with proteins, and usually one or more nucleoli, singular, nucleolus.
What happens in the nucleolus?
It's the ribosome factory, site of ribosomal RNA synthesis and assembly of ribosomal subunits, no membrane around it. Cells making lots of protein have prominent nucleoli.
And the chromatin is the DNA itself.
DNA wrapped around histone proteins, plus other proteins. In a non-dividing cell, it's a diffuse network. That 2 meters of DNA per human cell has to be incredibly well packaged.
And when the cell divides, this chromatin condenses into
Chromosomes, those distinct structures we see under the microscope during mitosis or meiosis. Each chromosome, after replication, consists of 2 identical sister chromatids joined at the centromere.
And the centromere's position defines the chromosome's shape.
Right. Metacentric, middle, submetacentric, off-center, acrocentric near one end, telocentric at the very end, helps identify them. Some also have secondary constrictions, sometimes forming little satellites.
One last quick mention, microbodies.
Small, membrane-bound vesicles containing specific enzymes for certain metabolic pathways. Peroxisomes are a common example, involved in breaking down fatty acids and neutralizing toxic substances.
Wow. Okay, that was a comprehensive tour inside the cell.
It really covers the highlights from these sources, and the main takeaway is clear. The cell is the fundamental unit. Structure and function all tied together.
Every organism built from these incredible little machines. And the diversity is staggering, but the underlying principles, the organelles, the processes, there's a lot of common ground.
Exactly. From the dynamic membrane controlling access to the ER and Golgi processing materials, mitochondria generating power, the nucleus directing everything. It's a coordinated system.
And understanding this tiny scale, as you said, is fundamental to understanding all of biology, health, disease.
It really is the molecular basis of life.
So to leave everyone with something to think about, we've seen this incredible complexity, this intricate network of structures and functions packed into a single cell, all working together to achieve independent existence. Could anything less than that whole package, that intact, complete cell, truly carry out all those essential life functions independently? What does that tell us about the minimum requirements for something to be considered alive?
It certainly emphasizes this staggering level of organization needed right from the very foundation of life. Something to ponder.
It's definitely food for thought. We hope this deep dive has illuminated the amazing world within the cell for you.
Keep exploring. It's a fascinating area.
Thanks for joining us for this deep dive.